So I wanted to take some time to kind of bring it full circle. We started talking about viruses, and I said I was going to mention viruses in the context of central dogma. It's a little bit tricky because what we're looking at with the coronavirus is an incomplete part of this central dogma. So again, viruses don't count as being alive, but they do have a pretty amazing ability to copy themselves. So all of this information here, this is the genome of COVID-19 virus. All this information is present in the genome, this tiny genome of the coronavirus, and it contains the instructions for making all of these proteins. And these proteins have functions for replicating themselves. So basically the most important elements of a virus, or the essential elements of a virus, is that it's got to sneak its way into a cell and convince the cell to copy everything that existed about it. So if there are proteins, the proteins have to be made. If there's structural proteins, if there are enzymes that have to accomplish functions, those have to be made. So all together in this viral genome, just a handful of proteins are responsible for doing that. So a number of these proteins in here are called NSP, or non-structural protein. So the structure of the coronavirus as we see it, I think a lot of time is spent focusing on the structure for good reason. If people are developing a vaccine, then that vaccine is going to respond to the surface proteins, so these so-called spike proteins on the surface. That's one of the most important elements for developing a vaccine. Having your cells recognize the virus before it gets inside, before it starts making copies of itself. So those would be the spike proteins on the outside, these structural proteins. But the non-structural proteins are involved in a little bit of the behind-the-scenes enzyme type of function. So in the genome of coronavirus, there's the spike protein, there's the envelope protein, the nucleocapsid protein, membrane protein. Those are more structural proteins, but then over here, lots of these non-structural proteins. And I found it pretty interesting. I located an article. I'm learning a little bit of this along with you as we go. So hopefully you find this interesting and not too far off track. So schematic of the genome there, and then a table here of what some of these non-structural proteins do. So I think that non-structural protein designation might make them feel like they're not so important, but of course these are essential to making this process happen. So NSP1 promotes cellular mRNA degradation and blocks host cell translation, results in blocking innate immune response. So this would be involved in basically stopping the cell from working for itself, and instead hijacking that and causing the cellular machinery to make the products that the virus needs to happen. And other ones, RNA binding protein. There's another one on here I thought was pretty interesting. So we got here NSP14, MTACE, don't know what that stands for here. Methyl transferase, there we go. So methyl transferase adds a 5' cap to the viral RNAs. Sound familiar? So this virus has to get its RNA read as though it was a eukaryotic gene transcript. So maybe we'll take a little aside here and remember what was going on. What does the RNA look like when it's ready for translation? Eukaryotic mRNA. Sometimes I refer to this as mature mRNA. So we have our strand of RNA, single strand. Under normal conditions, this would have been made as a copy of the DNA template, right? So it's got a 5' and 3' end, which end on this arrow is 3' end. So 3' end is the arrow head, the 5' end is the I don't know, the arrow butt. So part of that coronavirus, one of the enzymes included puts this methylated G cap on the 5' end. And that's part of the signal that says this is a piece of RNA that should be translated. And what is the molecule in eukaryotes? I guess in all cells, what is the molecule responsible for translation? So for translation to happen, we've got the RNA and the, yes, good answer, ribosome. Sometimes gets drawn as this. It has two parts, a small subunit and a large subunit. And it kind of clamps down on the RNA molecule. And it clamps down near the 5' end and starts moving down this direction. So again, this arrow, very helpful arrow. When I was a student, I didn't appreciate just how much of the central dogma happens according to these same arrow directions. So the RNA molecule was made in this direction, 5' to 3'. It also gets read in this direction, the 5' to 3' direction. So the ribosome's on this RNA and goes looking for what? What causes the polypeptide, the protein, to start getting made? That would be the start codon. So somewhere in here is the AUG start codon. And at that location, that would be the first, so I'm going to fill this in here pretty quick. The start codon is a sequence of 3 nucleic acids, 3 nucleotides on the RNA. And that contains the code for what will be the first amino acid to be put onto the amino acid sequence. So let's say we had one piece of this gene from the virus. We've got the methyl cap on there. We've got RNA. If you remember, RNA was the, let's see here, the viral RNA. The RNA was the native form of the nucleotides, the nucleic acid inside of the coronavirus. So that RNA is like ready to go. And it can be translated as being shown here. And to complete the process of replicating viruses, then this virus with RNA has to be made into new viruses with RNA. And so there's this RNA-dependent RNA polymerase, which takes the source of the RNA and causes it to get copied into new RNAs for packaging. So let's look at one of those sequences. So the I had a question today. Maybe I'll clear that up real quick. A student asked me, we were talking about the different names for these. So coronavirus. There was novel coronavirus. There is COVID-19. And there's this SARS-CoV-2. So coronavirus is a category of viruses. And actually, if we go back to this PDF document, this is a document from 2015. So there was no COVID-19 by the time this was written, but this still talks about SARS-CoV and outlines the arrangement of genes along the viral genome here. And so these are SARS and different related viruses. These already existed when we did, it kind of depends on what they get called in the public eye. There was the disease called SARS, right? So SARS stands for severe acute respiratory syndrome. MERS is one that has shown up. So here we have Middle East Respiratory Syndrome. So the SARS-CoV, that's severe acute respiratory syndrome. This is really the name for the virus. And it's named for the disease it causes. Associated with the COV is coronavirus, and 2 distinguishes it from what was similar but already known to exist in the human population. So this idea of novel or new, novel coronavirus is like shorthand for, hey, we've got a new version of this that the human population hasn't seen before, as far as we know. That relates to an illustration I showed in the virus presentation. So this is the relationship of coronaviruses. This is SARS-CoV related and SARS-CoV-2 related. So a specific version of this virus cropped up recently, and it's known to exist in animals. And a lot of these, you'll see we have a lot of bats. So bats are known to have viruses that sometimes jump into humans. It's relatively rare, but this is basically where we get sources of new viruses. They don't just originate in the human species. I suppose they could by recombining potentially, but sometimes we get them and they come from animal sources. So this related virus, some are known to exist in bats. These are a couple versions. They're very similar to what the humans have had. So these are the original Wuhan, China humans that got coronavirus, the new version. And the closest living animal to have a sequence like that is this bat. The red dots here, these are pangolins, which are whole different mammal organisms, but they've been vilified a little bit as potentially involved in this. They may not actually be the source and might have been like a secondary introduction from bats to pangolins. This one particular bat still kind of implicates bats as the likely source, and then kind of bats all throughout. So a little farther distant, this bat also has a similar virus to coronavirus 2. So to find these, basically they're epidemiologists, people who study human diseases, might be surveying wild animal populations for the existence of viruses we haven't seen before. So maybe allowing people to prepare for something like this to happen in the future. So this is kind of a genomic start for where coronavirus came from and the origin of this new name, SARS-CoV-2. The other part, COVID-19, is a name, more of a name for the disease. I guess our current, we'll call it a pandemic. And that's just coronavirus disease was my understanding. So, oops, sorry. Stop. Coronavirus disease, and the 19 for 2019 when it was first known. So it's certainly boomed on the scene in 2020, but it was known at the very end of 2019, and then it got this name COVID-19. So if you're familiar with AIDS and HIV, that's a similar relationship where the virus is called HIV, the human immunodeficiency virus, and the disease is called AIDS, the acquired immunodeficiency syndrome. So here we have the virus SARS-CoV-2 and the disease COVID-19, and then just sometimes called coronavirus in the common literature. But of course, calling it coronavirus doesn't really set COVID-19 apart from other coronaviruses. So COVID-19 is a specific name, SARS-CoV-2 is a specific name for what's going on right now. All right, back to our structure here. where were we? Right. So we were looking at, this is a resource run by the National Institutes of Health. Lots of scientists all over the world are researching coronavirus right now. And part of researching it is to run sequences of what the genome looks like. One potential problem could be if this thing mutated over time, right? So changes in the nucleotide sequence potentially could cause coronavirus to mutate into different forms. Some that might escape the immune system. If somebody's already immune, developed immunity to the first coronavirus, they could encounter a new version that's not susceptible to their immune system or not recognized by their immune system. So it's important to kind of keep up on the changes to the old coronavirus as time goes on. That's something I also talked about in the virus lecture. So this idea over here of changes over time. There have been quite a few changes, but my understanding is there's not been a whole lot of, there's not really been any change that looks like it would affect our ability to develop a vaccine or become immune to this. Although immunity to a respiratory virus, as I understand, is also a bit of a challenge. So we will see how the old virus development proceeds. All right. So this is basically a cumulative summary of lots of genomic sequencing. Sequencing the nucleotide genome of a virus is relatively inexpensive and fast these days. Certainly laboratories are funded to do this because the world wants to know, right? What's going on with the coronavirus, and is it changing to something new that we need to be aware of? So I just did a search on coronavirus DNA sequence or something like that. And there are a variety of different resources here. PubMed might be publications about coronavirus, so in the literature. And then nucleotide records, these would be actually sequences, nucleotide sequences for the genes of the coronavirus. So we can just jump on one of these, released today. Wow. So this is actually today as I'm recording this. There's a new block of sequences from the United States, Virginia. Just, I don't know, kind of blows you away. Look at all the different sequences. Lots of research going on here. Okay, so this will get us to a record of the nucleotide sequence. And I wanted to kind of highlight that as well. So if we scroll down, this one is counting off the nucleotide letters. I think it's just maybe by convention as much as anything, these are given as DNA nucleotides. So I don't know if the method they used got them DNA nucleotides or if they just converted the RNA nucleotides into DNA. But of course, anytime you're looking at a sequence, if you see the T's instead of U's, that would be a DNA sequence. If you see the U's, that would be an RNA sequence. So big blocks here. Let's go back to see if we can relate these two documents together. So this is the schematic of the genome. And we had that block here of what some of these non-structural proteins are. Here's one I kind of wanted to do, a hexadecimeric complex of NSP7, NSP8. So let's see if we can maybe find NSP7 and 8 and see if those are present on this one. Sure hope so. NSP7, there we go. Maybe. All right. That looks a little challenging. So I don't see, what I was looking for would be a start codon. I don't see one initially, and I don't want to hunt around for it right now. I'm going to go to one that has a little bit cleaner presentation there. I think, I don't know, let's see if we can figure this out. Okay, so, well, it's possible here that we've got a Yeah, so polyprotein. I believe this is translated as one protein and then maybe carved up into protein subunits after translation. And so among the products would be these various peptides. That's a little bit out of what we've talked about, but definitely a possibility. And viruses, of course, would be playing by different rules potentially than either eukaryotes or bacteria. So let's go with one here that's got a little bit cleaner presentation. These polyproteins, they have an amino acid sequence, but they're darn big. So let's go to something that's a little bit smaller. All right, here we go. The S, the spike or the surface glycoprotein here. So here's a gene for the S product. This would be the gene portion of that nucleotide sequence that encodes for the spike protein. Oh, it's a big one. Let's see what I can do here. S display. Try to get it. That looks too small. Nope, I think that might work for us. All right, so I wanted to get it into a form that didn't have the numbers there. Let me. That's not gonna work. Sorry, we're winging it a little bit here. All right, so surface glycoprotein. Yeah, I think that's right. So I copied that, and it gave us a translation. So a couple things to notice. The part that's highlighted is the code determining sequence. So it's basically the part of this nucleotide sequence that directly contains codons that will become the polypeptide or the protein. And as such, it starts with an ATG, right? So ATG, we remember on RNA looks like an AUG, that's a start codon. And the start codon corresponds to a methionine amino acid. So it goes from start all the way down to something that is probably a stop codon. Let's find out. So I can do a search for RNA translation. See what we get here. Translate tool. Let's try that one. All right, so it turned into a pretty big sequence. I'm going to paste in. So it starts with ATG. Again, just sort of by convention, scientists know that DNA can be converted into RNA. Here's a question for you. Are we looking at which strand of the DNA molecule are we looking at? Sorry, side break. All right, so if we have a DNA strand, let's see how this starts here. ATG, TTTG. ATG, TTTG. Is a piece of DNA, and the convention is to write that 5' to 3'. Then on the DNA molecule, let's forget it's a virus for a moment. On the DNA molecule, it would be part of a double-stranded piece that has complementary nucleotides and a backbone that's running in the opposite direction, or anti-parallel. So anti-parallel means it's running parallel to the other sequence, but in the other direction. So all of these nucleotides are making base pairs, G with C, A with T, G with C, and so on. So we're looking at the DNA sequence, but then after translation, we would be looking at, again, by convention, the sequence that's given here is the coding strand. So this is the coding strand. otherwise known as the non-template strand. And it's complementary to the non-coding, otherwise known as the template strand. And so, I'm sorry, cross that out. Transcription would get us a molecule that runs the same direction, 5' to 3'. Let's get rid of that. And has the same sequence as the coding strand, except with U's instead of T's. So when we look at the sequence of DNA, you can just mentally substitute in a U instead of T, and that gets you the sequence that would result from the RNA molecule. All right, let's see what happens here. Translate. Okay. So this website, expasy.org, gives you a lot of options. What it's doing is it's investigating different reading frames in different directions. So we pasted in something that we knew was in the right reading frame, and also was running in the 5' to 3' direction. So this is what we got, MFV, FL, and so on. Started with an M methionine, ended with a dash. That's probably what they did for a stop codon. And it's a continuous sequence throughout. So this is one continuous sequence of amino acids. Each letter here is the single letter abbreviation for an amino acid. And these other options are not what happened, but maybe is a nice illustration of what a different reading frame would get you. So they get you basically garbled up nonsense if you are trying to translate that sequence anyway, except for the way that it should be translated. So there's really not a second best translation. It's either the right translation or complete nonsense. So we can compare this back to the translation that was provided. So MFV, FLV, back here, MFV, FLV. All right, so that's the same. So this is the protein sequence for the spike protein. We could also, if you wanted to take this sequence, could look at the sequence and find out what the codons are. You could take this to a codon translation table. And let me put this in a different window so we can use it. All right, so we find MFVF. So ATG is AUG, that's our start methionine. Next up, shoot, TTT, or becomes UUU, that's phenylalanine. And starts with the F sound, so that's one way to remember. Phenylalanine is actually abbreviated F. Next up, GTT, GUU, valine, that's V. Here's another TTT, also phenylalanine, F, CTT, CUU, leucine, MFVF. There's another leucine. Right, so you could do this by hand, but it's also easy enough to teach a computer how to do this. So this is a computer reading this sequence and telling us what the amino acids should be. Let's look at the last couple codons. So second to last, ACA. Here's ACA threonine. That's a T over here. And the last one, TAA, becomes UAA. That is stop. And if you remember from what we covered in translation, stop codon does not have a corresponding amino acid. It just brings in this release factor and causes the amino acid sequence to end. So that's the end of the polypeptide. So it goes from M-methionine to start all the way down to threonine as the last one, and then it would terminate. And then you're left with this giant sequence of amino acids. Then they fold around each other and get you the functioning protein, ideally. Let's look into this hexadecimeric complex again. So we had a little trouble getting the translation there because it was translated and then cut. It was a little bit complicated, but we could look at this resource here. It says these form a hexadecimeric complex, reference 143. Do a little search on that. So I'm going to copy the title, and I'm going to go over to Google Scholar and search on that, find it there. And this tells us something about this. So again, this is 2005. These are proteins that are found in the novel coronavirus that causes COVID-19, but they're related to proteins that have been seen before. So this caught my eye because as a hexadecamer, what that means, the hex for 6, the deca for 10, so this would be, I believe, a 16 subunit protein. So we talked about protein structure last time, and the idea that some functioning proteins are, in fact, built up by combining the products of several genes together. So in this diagram, they've got NSP7, NSP8, and 2 versions of NSP8. And they're colored differently. These are relatively small proteins. And I don't know if we can count them all, but there's a, I don't know, looks like at least 2 orange, 2 green, and some number of blues in there. Altogether, that forms the complete protein structure that can do the protein function. And the gene sequences for those, the nucleotide sequences, contain the instructions for making the proteins that then merge together, and then ultimately this thing has the function that was described over here on 43. Go back, there it is. So processivity clamp for RNA polymerase may act as primase. So it's an enzyme that has some function that, as of this writing, was not completely worked out. So it's a big world out there. You might get the impression here from digging more into this. Even as something with a genome as small as a virus, there's still lots to investigate there, and lots of important avenues to investigate. So if a virus is going to have a way to be extra virulent, you know, attack cells even more aggressively, they might be related to some of its genetic components. If there's a way to inhibit this virus, keep it from being as virulent, then some of the answers might lie in investigating the proteins and what they do. So sort of learning about our enemies is a valuable way to prevent being negatively impacted by them. All right, so I think that's probably a good summary. I don't know, I wanted to kind of jump in and talk about translation, show you a connection back to the viruses. One other connection I think I mentioned with the, in the virus talk, this idea that you could potentially develop a vaccine using an mRNA starting point. So this idea of a nucleic acid vaccine, knowing the sequence for the gene that makes the spike protein that we just looked at, you could inject, rather than injecting parts of an existing virus, into a cell. You could teach a human cell to make these viral particles. So what would you want to add? You'd want to add the RNA, or kind of on a molecular level, teach a human cell to make that RNA that would end up making a spike protein from a human cell. So as far as potential damage, if a person's cells are manufacturing just a spike for coronavirus and not any other parts, then that's not anywhere near manufacturing a virus. It's not any danger of contracting coronavirus, just a tiny little bit of coronavirus. But these viral spike proteins can end up teaching the immune system what to look out for. And of course, these would be the proteins that are on the surface of the the virus as it comes into the body. So an interesting, apparently not used before, but if it works out, be a really interesting way for preventing or preparing people with immunity to viruses like coronavirus. All right, let me stop there and see if there's anything else I want to add later.